Welcome back to this to the session 2 with some panel discussion. And now we have the moderator, Joseba Pepa Anton, who is a professor of microbiology at the University of Alicante and head of molecular microbial ecology group. Her research focuses on the study of the microbiota of hypersaline environments and marine sediments, as well as study the marine invertebrate symbionts using metagenomics, as well as other molecular techniques and culture-based approaches. So please.
Well, first of all, thank you very much. I have to say that I've been advisor in the SIMBA project, and that has really been an honor. And also, I think I receive way more than what I given. And I think it's your achievements have been really remarkable. And we must consider that you have got all of these results going through a pandemic that we all have fortunately forgotten somehow, but you had some problems related with this, and I think this is an extra challenge for the whole project. So thank you very much, and my congratulations for your achievement. And so we have now in this panel on microbes, food, and human health. And the first thing we will have is the introduction of our panelists. And I must say that the previous moderator set the level very high, but I will try my best anyway. I don't know even where I should put, because you did fine. So anyway, so you can start with, we have 3 panelists here and 2 more online. So please go ahead.
My name is Anne Salonen. I'm from the University of Helsinki. I work on human microbiome, and I'm a director of the human microbiome research program we have at the Faculty of Medicine in the University of Helsinki.
Thank you. My name is Zsófia Kádár. I'm coming from Biobase Europe pilot plant, and the ones who were not involved in the project, this is a pilot facility for upscaling in Belgium. My role is project acquisition coordinator. That basically covers that I'm responsible for public projects at the Biobase Europe pilot plant. And I'm very happy to be here. Thank you.
Thank you. So my name is Maher Abu Hashim, and I'm a professor at the Department of Biotechnology and Biomedicine here at DTU. I have a bit of a different background because I have been working, I'm a protein chemist by training, and I have been working on carbohydrate-protein interactions throughout my career. And maybe since 2010 or something, I got very interested in the interaction of our gut microbiota with both dietary and host-derived glycans. And this is what I've been focusing on, doing a little bit more microbiology and other cross-disciplinary approaches to understand this.
Okay, thank you. And Lorena? Can you introduce yourself?
Hello, everyone. Yes, thank you for the invitation to be part of this amazing panel. So my name is Lorena Savani. I'm the mission lead of Healthier Lives Through Food in EIT Food, which is a knowledge and innovation community that is dedicated to making the food system more sustainable. And actually, I'm supporting with projects and collaborations to make a better, healthier food system. I'm also working in the think tank of protein diversification, which as well as there are some aspects about here that we're going to cover today about plant-based. So my background is a food engineer, and I have been always working in innovation in big corporations and now in EIT Food since 2018.
Thank you. And finally, Nelson.
Okay. Well, good morning. Thank you very much for inviting me to this very exciting conference. My name is Nelson Mamedori. I am a professor emeritus of environmental biotechnology at the University of Parma, where I have been for the last 20 years full professor in biotechnology. My role within the SIMBA project was mainly on the exploitation of microbial controls for improving the health of the soil, for improving the quality and the production of the plant. And my background is genetics, and well, I am unhappy not to be there, but I am jumping from one lecture to the other in this day with my students. Thank you.
Yeah, we understand that. Thank you very much. So, as you know from the talks, this is like a multi-side problem in a way. So we start from microbes involved in food production, but in a way, microbes are also like the end users somehow because they are part of our microbiome. And of course, there is also an effect direct on it may be an effect on our health. And in the mean, to make this more complicated, we have to bring it to the market. So we have like different aspects on the whole process. The food, the microbes, either as food makers and food consumers, and the market. So to start with, Nelson, I think we can start with you. How do you view now the application potential of the food, microbe-based food in our market now? How do you see, in a realistic way, how do you see that there is really a great opportunity for developing and buying and selling this food?
Well, there are two levels in which it has been said. Microorganisms can be applied across the food chain. There is a level in which probably the certainties are more, and there is a level in which the certainties are less. The first level is in the field. In the field, the application is going on since some years with some interesting results, and both in terms of safety, quality, and yield of some agricultural growth. Going from the field to the processing industry and to the commercialization of food, the gap is the gap probably is increasing, even if the technology are more commandable and more developed. But there are, I take as an example the Italian scenario.
Can you repeat it? An example of?
Yes, I am using as example the Italian scenario because it is the one that I know it better, and I don't want to make mistakes. But for the Italian scenario, there is a need of increasing the use of biofertilizer in a way supplementing the chemical fertilizer or avoiding the large use of chemical fertilizer by reducing the amount, in this way also, the amount of greenhouse gas emission, which is one of the plug of the intensive agriculture. I have made some calculation that about 80% of the emission are due to chemical fertilizer. By replacing only 50% of the chemical fertilizer by biofertilizer, we will have a saving of about 40% of greenhouse gas emission.
Okay.
This is particularly interesting in an environmental sense. Unfortunately, if we go from the environmental side to the cost side, to the economy, the advantages do not exist.
Okay,
The use of chemical fertilizer is still less costly than replacing the chemical fertilizer with biofertilizer. Something, of course, completely different exists in the processing industry. In the processing industry, where I can see a more promising role of microorganisms is in the recovery of industrial waste. Always taking the example of the Italian industry, the basic processing industry, there is a huge amount of waste that actually are not exploited, that with the application of the proper microbial technology can be transformed in good both for the pharmaceutical and also environmental purpose. And this is something that interests me more than the application of the standard microbial technology to food production.
Okay, thank you very much. So you really see the need and the opportunity. So why is not being done? Why are we so far using these products now? What do you think the drawbacks are that are holding us for really implementing? What do you think?
Yeah, I mean, just my personal opinion. Probably it's the acceptance from the people that can be one of the barriers that are not better known, and also people might be afraid to use this kind of products in daily basis. I do not know if someone else has another idea.
So I think probably one needs to, or it might be good to distinguish between products which are for the general healthy population, which are for maintaining or preventing disease or maintenance of health, and the use of such products for therapeutic purposes, where you want to address, for instance, people who lost their microbiota during antibiotic exposure or things like that. So probably the products need to be more refined. So that's one potential thing. Another thing, which in my opinion is major, is that we still lack understanding of the interaction of these products with the microbiota. Mm-hmm. So informatics gives us tremendous information about which microbiota groups are there, at which time, etc. But the underlying function remains largely lacking. We are not good in predicting because there's several layers beyond the potential to do a certain function. There's some regulatory aspects. So a bacterium can express several pathways, but we don't know which are expressed under which conditions. We don't know also when a certain food presented, how the presentation of the food product interacts with the microbiota. So some microbiota are able to adhere, for instance, to insoluble particles, and they are able to process these complex things, whereas the vast majority aren't. So there's a hierarchical ecological system which we don't know much about. We know, for instance, that two bacteria can use structural polysaccharides, like from grains, xylem, but we don't know if they are presented a cereal particle or an oligosaccharide, who will be more competitive. So I think we need much more functional knowledge. And there's also a lack of knowledge on the capabilities of the microbiota, because 30% of the genomes are unannotated. So our level of insight into what the microbiota are able to do under a certain condition, predictability of that is still lacking, and there's some work to do there.
Mm-hmm. So there seems that there is like at least two aspects. One has to be with the social aspects that were discussed this morning, as you said, like how the end users, the human end users, see the thing, and the other is the science on it, and that seems extremely complex. So Lorena, what do you think? What would be like?
Yeah, I mean, from my point of view, I think the microbial processes have an immense application potential in the food industry, actually, because it offers a very innovative way to produce nutritious foods and feed as well, efficiently and sustainably. And actually, we are seeing now growing this dramatically using microbes to ferment plant-based ingredients that can create dairy alternatives like yogurt or cheese. And while at the same time, there is also the sustainability aspect associated with that. However, I agree, as someone mentioned as well, that there is a kind of some restrictions and challenges that we need to face to be able to provide these solutions into the market. And one of these is really the consumer acceptance. So this is something that we still need to work on. And the scalability. For me, in the industry, as for an industry point of view, scalability to scale the production is one of the biggest hurdles, and also the ensuring the consistency of the product quality. So this is something that still needs a long way to go, while at the same time there are some regulations as well that need to be addressed. But yes, this is, from my point of view, how the biggest challenges. And also, I would also mention the affordability of these new products. Of course,
sorry, I didn't.
As far as they are not scalable, yeah.
Mm-hmm. So who has to do this? Who has to bridge this gap to understand what consumers need and to convince them? We will go to science afterwards, because who has to do this? The European Union, who's in charge of this? Because you were in the work that was presented this morning, they were saying that it was in the case of the farmers, they were not very willing to trust like the government institutions. So how can we manage to convince people if we have to? I don't know who's in charge of this, or whom should be in charge of this. Do you know? I don't know. Maybe someone in the public now, just raise your hand. And
I think here would be very important to have an effective communication and education, which are crucial to to convey the benefits of these type of products that are being brought into the market. And then of course also the aspect of the regulation is a key aspect to be addressed to be able to bring those products into the market faster.
Mm-hmm. Okay. So, and one of the other sides, which is, you wanted to say something? Go ahead, sorry.
I just wanted to say that it seems that people trust scientists more than politicians.
Well, yes.
So maybe scientists have a responsibility, and they have integrity also to communicate this to the public. That requires, of course, robust work underpinning these products.
And you don't see a problem, because normally science are not very willing to say that something is 100% sure or 100% certain, and this may be a bit confusing, or you don't see a problem in this?
I mean, so I think that's true because on the other hand, the credibility with the public is very important. So because they get exposed to a lot of conflicting reports and to so I think it's important to try to have at least a scientific community try to communicate to the there will be not 100% if scientists do this, but at least it will be hopefully more effective.
Just a quick comment coming back to you mentioned earlier, and I very much agree that their segmentation, whether you target healthy individuals or those ones with disease, is important. And actually, from the regulatory point of view, it is of course a key aspect, because if you want to prevent, if you work with organisms where you want to prevent a disease and your target population is healthy, then your microbial products are, you go for the food supplement route. But as soon as you target the diseased population, you are with the medicinal product and you are working with the drug. So, yeah, that we have.
And so going back to the first aspect of the microbiome, I think this is something key that has to do also with what you're saying. Do we really know what a normal microbiome is, so we can tell the changes induced by diet or something? Or what? Can you please tell us some?
Yeah, it's been really far from trivial on defining what all is a normal and healthy microbiota, but I think especially in Western populations which are quite kind of well sampled and analyzed, I think we understand the variation which all can be considered normal these days much better. But of course, because of this huge variation also within healthy individuals, then it becomes, that's the major challenge. While the individuality of the human microbiota is also a kind of, where we can really, there is a lot of potential when we talk about individual responses to diet, etc. But from the research point of view, it's also a major challenge because basically you have within your, if you, for example, carry out dietary trials, you typically have within your one group, you already have so much variation in the microbiota that it makes it much more difficult to find statistical differences than between your groups, for example, for treatments.
And is it possible that you still have the same microbiota, but different, very different activities that you wouldn't be able to monitor?
Definitely, yeah. There is a huge functional redundancy in the gut. And that's maybe I use this opportunity to kind of clarify one point regarding the microbiota diversity, because we are kind of taught to and adapted the idea that the higher diversity in the human microbiota, the better it is. And that's of course true still from the perspective of the ecological resilience. But what we also now know that if certain types of microbial fibers are fed to individuals which are only used by a selective group of organisms, it's not uncommon that you actually decrease your diversity, so the kind of number of taxa, because you specifically boost even, for example, some butyrate producers which are good. So it's not so simple that sometimes, for example, by feeding resistant starch, you may actually see that the diversity, the alpha diversity goes down. So that's good to keep in mind also.
Well, actually, I think that even in the media, there is an idea that decreasing the diversity is bad. I mean, something that is getting like common thought in. So would this be the case? Or?
So, I mean, of course, if it goes to that level, for example, after an antibiotic course or repeated antibiotic courses, that you decrease the diversity so much that many of the core ecosystem functions are lost, then it will be a detrimental thing for your health. But I think we have to be more specific when we look on the dietary effects on microbiota, that not jump into conclusions that or come with the claim regarding the alpha diversity that in order to be beneficial to human health, it should always increase the diversity. That's exactly for the reason I just explained, not always the case. So we need to look for specific organisms that produce beneficial products, for example, from dietary fibers.
Mm-hmm.
So I think this is a very important point because I think our knowledge about the microbiota is derived mainly, our functional knowledge, from abundant generalists, whereas the majority of the microbiota are different specialists, and these specialists are underpinning the inter-individual differences, and they are more vulnerable to erosion. So there has been an erosion of the microbiota due to westernized diets, and some of these groups have been lost at certain individuals, and they differ a lot inter-individually. So I think in that context, probably one needs to think of not the most simple products that can be made, because like a simple product, which is a resistant starch or something like that, or xylan, or would hit one group, and then you would sort of suppress many other specialists. Some of these are performing key functions. So I think one needs to attend this by trying to have more complex products that maintain this diversity and which are directed to more specialist bacteria that are differing, but some of them are keystone with regard to the functions and the substrates that they convert.
But there seems that there is a very difficult to solve problem, because we want to go from this fermented potato starch or whatever to the market, and if we have to find out for every product these nuances, how would you solve do we need these very exhaustive studies to try to you are more on the biotechnology, what is your feeling? So because if we want to sell, to end up doing something, we will have to solve things.
I can reflect mainly on the scalability that has been mentioned by Lorena at the beginning of the session, and also at the end of the first session, when it was the message, what would be the message for the new generation, it was scalability. And because this is our key expertise, this is the service that we are providing, so we can actually see many times, that really good ideas that are developed in the lab, they are not necessarily scalable. And that's really a pity, and we are facing, a lot of difficulties when we have to scale up these processes. So I think this is indeed, that has been mentioned, this is one of the key aspects that everyone has to keep in mind, how the process can be scalable. Therefore, we like to be involved also in the development phase, usually in the projects, to have the discussions, to see from our point of view as an expert in upscaling what is feasible. Sometimes it can be challenging technically, that we also experience within the SIMBA project, that we had difficulties in upscaling the fermentations because of certain elements. And sometimes, what is feasible in lab scale, in small scale, it's not feasible in bigger scale. For example, considering safety aspects that are not present in the lab scale, but we have to consider in big scale. And also the other aspects, actually, especially when we are coming to food, to how we keep all the food regulation in mind when we are doing upscaling. So that also brings a lot of effort to upscale a process that is related to food. A lot of regulations that we have to follow, we have to keep in mind and take into consideration when we are upscaling a process. So I just wanted to reflect on that point, that this is really a key element, and I'm very thankful for Lorena that it was her who mentioned it.
So specifically regarding the regulations, do you think that the legal framework in Europe is adequate and it's, updated enough to face this process, or we would need to have new regulations to implement the knowledge about the microbiome, all the processes needed, all the quality checks? What's this? Because it was mentioned before about sometimes regulation goes behind the findings. So what's your opinion?
I'm not familiar with the regulations regarding the microbiomes itself. What I can see also from other European projects that it's general, there is always a bottleneck with the regulations. Whenever we talk about microbiomes, bioplastics, it's always a topic that it's coming back, that it's really missing, especially when we are developing something new that we want to exploit or bring to the market. Regarding the food process, when we are coming to upscaling, it's very well regulated. Also in our company, we have a quality department who are there for checking all these regulations and follow them. So I think that part is well covered, but is really missing the regulations for these new products that we are developing.
And Lorena and Nelson, what do you think? Do we need to change like the framework to be able to speed up the process of?
Yeah, with what regards to regulation in Europe, unfortunately, we have a much more strict regulation compared to other countries, and like I don't know, Singapore, Canada, US, and therefore the implementation of these type of products that are coming from these new technologies are much longer, especially when it concerns to novel foods. When we are talking about novel food, then there is a huge process that you need to undergo to get the approval and you need to present a lot of documentation and safety as well. The documentations to ensure that this process is safe and therefore it's a long process. So if we compare with other countries, it's still.
Mm-hmm.
Yeah, if I can add something to this, well, I find it quite obvious that regulation is always behind the scientific discovery and the technological progress. It cannot be different from this, of course. And by the way, I also believe that this is a safety issue, that the society is always taking to protect the citizen in a way. The problem for regulation and for acceptance of new technology and new discovery, it has been mentioned, is the consistency. And the consistency is very important both for the regulators and for the public. The problem in this field is that sometimes the consistency is not, I would say, communicated. The consistency of the results and of the achievement is not communicated properly. So this determines a sort of confusion in the public and some, let's say, constraint in the regulator. And these are the two things that mostly generate, let's say, problems in this field. But there is nothing that cannot be overcome. I want to be positive. Okay, so the scientific research is going on, acquiring more certainty, producing more consistency in it, and obviously also the regulator and the public will be obliged at the very end to take note of this in the proper way.
Okay, thank you very much. And well, I think we are running out of time, but one more question. In this field of microbiome and microbe-based solutions, do you see something outside Europe from which we could learn in the UA or the other way around? Can we learn something from how these microbe-based foods are being implemented or have been traditionally implemented in other parts of the world? And the other way, what can we bring? What do you think? There is something outside Europe that we should be learning? I mean, for sure there is something we have to learn from, sure, but something specific in this field of the food-based, microbe-based food.
Last year it was one of the conference at the EFIP, but it was one of the topic exactly that how Europe could look out and what to follow, and what was mentioned exactly the regulation to make it more flexible, much faster. It would help a lot of development also to get onto the market earlier, because we're also experiencing that there are some companies doing the upscaling, and whenever they reach the level that they need, they do not find the possibility to go onto the market in Europe, and they go to the US, they go to Asia, because it's just much easier, because it's very slow process to get things changed here in Europe.
So you are speeding up, like for the CRISPR's modified systems in general in Europe. So you think that the machinery, the legislation is too slow?
It takes. This is what they also gave a presentation, how long it takes the process itself, all the paperwork and all the process, that maybe that could be shorter.
So do you all agree? Oh. Briefly, because she's looking at me and saying...
I think there might be some cultural aspect. I mean, in Japan, for instance, there's a lot of the traditional food which has a lot of fiber, which has very, very high nutritional value, and there's a much broader accept to it. And so I think maybe the consumer aspect is important to work on, because this has been considered a bit like, for many years ago. Now things are changing, hopefully, and there's more accept for this type of products.
There is like a fashion now on fermented foods and kombucha and all these things. So maybe it's a good moment now for this. This is the moment, maybe. So I think we already finished. Thank you very much to you all. And I hope you enjoy I'm sure you enjoy it's a shame that I couldn't talk more. I'm a microbiologist, so I would have spoken more about microbiome, so I refrain myself. So thank you very much, and so.
Thank you.
Thank you.
Okay, questions now?
That was a very interesting conversation. I'm just wondering a little on the herbal medicine side, and you mentioned Japan and you mentioned, Asia. I'm wondering if there is many of the practices, or at least the principles that are applied in herbal medicines, that could be a little, if you like, translated over to the microbiome. Because in the regulatory side, there's a lot of herbal medicine expertise already there. So, with the herbal medicines, it's not just one bioactive that's present, but many different bioactives that are present. So that's just one comment. And the other comment is, can we envisage a little menu where I say, I want a good heart, I want to be slim, and so I choose this menu of items, and if I want to be, maybe faster, or, I want my hair to have a lovely glow or something like that. Can we envisage a future like this for these types of products? I don't know. It's just something that struck me as you were talking, that, this might be a possibility.
So who's going to answer this difficult question?
I cannot answer for herbal medicines, unfortunately, but as a general comment to your second question, that while the gut microbiome is being studied a lot for its role on explaining and driving the individual dietary effects, there is, I do see that, especially integrating with the host genotype in the future. I mean, because we know already in practice that the health benefits or risks coming from identical food are not same for everyone. So I think we are going there, that, for example, based on your, I think, host genome, but also on your gut microbiota, that you know that you should, for example, not to get too much gas, but still get the benefits from fibers. Maybe you should go for this. And some other people are maybe getting more potential carcinogens from meat because of the gut microbiota. So in the future, yes.
I think we have a question. Were you going to say something, Ma?
Just a short comment on your herbal medicine. I think this is a very important question because the microbiota actually are our pharmacists. So the bioactivity that we get is a function of the interplay with the microbiota rather than the things we take in per se. So that's very important to attend, and there's some knowledge about that in the field.
I think we have a question from Thomas. Please go ahead.
Yeah, thank you very much. It's very interesting to follow these panel discussion. I'm from the university in Jena in Germany. My question is about the discussion about diversity. So again, in this session, and we are discussing always alpha diversity, but I think it is very important to focus more on the functional traits. Are these functional traits constant during changing microbiome? And this kind of stuff should be investigated more. I think just focusing on a changing microbiome does not help us to make also a good risk assessment or to understand the beneficial impact of a microbiome on a host, for example. So a changing microbiome can have still a constant functional trait in total, or changing microbiome can also have constant features providing to the host. And I think this needs to be addressed much more carefully in the future. And maybe somebody of the panel likes to comment on that as well. Thanks.
Just a quick comment. Thank you very much for your important comment. I fully agree. We were optimistic that we could just based on the composition, microbiota composition, because still certain functions are limited to specific group of taxa, that we could use those as an easy, cheap measure, but it's exactly like that we have to go for the functions. We've moved to counting genes on metagenomes, but even that, as you say, the expression in the end might be that differs, and that's why looking at metabolites, looking at proteins, also transcripts, which are in the fecal matter, they are not so relevant, but I fully agree with you.
I think this is very important to also for a risk assessment in the future, because we need to know what's actually happening in the chemosphere of such a microbiome. What is actually released, produced in the interaction between host and microbes, and I think a microbiome and even a metagenome analysis is not really telling us this story.
Thank you very much. And I think we have finished, and I really appreciate all your participation. Thank you. What's this? An umbrella. Thank you. Very useful. Yeah, tomorrow will rain, I think. And I didn't bring one. Thank you very much.